This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 73, recorded on February 20th, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, everybody.
How are you?
I'm okay.
Just okay. You haven't gone anywhere. You're just sitting there in Ann Arbor.
Well, I'm teaching right now, so I get to spend Monday and Thursday morning with our bright graduate students.
What are you teaching? What class?
It's a pathogenesis course, so it's literature-based. We discuss a couple of papers, not unlike TWiM.
This is a graduate level course?
It is.
So you have, 10, 20 students?
15 or more.
Nice.
We draw from a couple different programs.
And they're all bright.
Of course.
Just like everyone in Ann Arbor, right?
Yep.
Amazing.
And polite.
That's great. Well, I'm teaching my undergraduate course at the moment.
Oh, good for you. How many?
190.
Wow.
That's a lot.
Yeah, it's a lot. But we just had our first exam of 3 on Monday. And we will probably lose some. The drop date is Tuesday. And we will always lose some by then. But I enjoy it. It's a lot of fun. It's a big room, and it's all lectures because that's all we can do. But I record them. They're all online, and a lot of them can listen over and over and over. So it's fun. I like it a lot.
On the exam lecture, is it essay exams, short answer problems, or multiple choice?
It's a mixture of short answers, very short answers, and multiple choice. We tried longer answers in previous years, and it's just too much for this number of people.
Yeah, I struggle with consistency and grading, and I've heard all the discussions about rubrics, but fatigue just sets in.
Yeah, it's hard
after a while, and to be consistent in your grading is just so challenging.
We made up a new kind of multiple choice this year, which is you have not just a short sentence, but a couple of sentences saying something, and then a sentence with different choices embedded in the sentence. This means that blank, 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 or blank, And that's a little better than the traditional medical student multiple choice, if you know what I mean, Michael.
Yeah. Well, the National Board of Medical Examiners has experimented with all variants, and the psychometricians have come up with the single one best answer format is the best one, but they've now gone to the triple jump, and it sounds like what you've done with yours is it's really a triple jump where you need to know 3-4 facts in order to get the correct and best answer.
Yeah, that's right. What I find weird is that we're still using paper.
Mm-hmm.
And because you can't give them computer-based exams. I mean, you can't let them take it at home because they'll cheat. It's unfortunate, but they will. They'll collaborate, which in my view isn't bad, but people don't like that. Or if you give it to them in class, they have their laptop screens up and everyone looks over everyone else's shoulder. So this seems crazy to me in this day and age. I have to print out 190 exams, right?
Mm-hmm.
Schlep them down there on the subway, then schlep them back up again, and then it's just I don't get it. But
well, what I did when I was doing a course similar to what Michele has been doing, and I did this for a number of years, is I gave them 30 essay-style questions a week before the exam, and I typically had a class size like Michele at Michigan, where they were 15 to 20 graduate students on average, and they referred to the questions as Schmidt's problems from hell. And
nice.
Because there was no way one individual would be able to solve all 30 of the questions effectively. And so what I told them to do is to work in groups and discuss the questions amongst themselves, divvy them up. And then on the day of the exam, I would go in and pick 5, and they would sit and write cold. And, with 15 graduate students, you can effectively manage And my colleagues always said to me, aren't you all going to get the same answer? Aren't they just going to memorize their answers? I said, no, because they are writing them cold. They don't know what out of the 30 questions, because they were similar but different. They all covered the same sorts of learning objectives associated with the course. And what I learned from this process is the students, by virtue of the fact that they were studying with each other and they were arguing the science, almost like what we do with TWiM here, where we discuss the paper and we tried to work through a particularly challenging point, they were doing that with each other. And so consequently, what I would do is often get very insightful answers. They would polish their prose, and it turned out to be a better experience than just showing them an exam question cold and then hoping for brilliance.
Yeah, it's nice.
So it worked well. And I, too, am teaching this semester, except this is I have 81 of our dental students, and they just had their exam last Friday. So
they're pulling their teeth out, huh?
Literally. But they reported it was a fair exam, and this year, because we had 2 snow days. Now, I mean, that's unusual for South Carolina. We had 2 snow days before the exam, so this year's average was 10 points higher than last year's class. And so they're now all boasting that they're smarter than the sophomores.
It's higher because they had more time to study?
Yeah, they had no class, and so they didn't. Typically, our dental students are very busy during the day with lab activities and simulations. And so 2 days off where the school was physically closed and we weren't letting them come in, they were able to stay at home and actually study.
Wow.
And so it actually is reflected in the material. And I, too, like you, we record our lectures so that they can go back and review them over and over again, because we're trying to cram a lot of information into our students' heads, because with each passing month, another batch of journals comes out.
Sure. In fact, less than a month.
Yeah, less every 2 weeks for some of the ASM journals.
Yep. Okay, let us move on to our paper for today. It is an article in Cell Host and Microbe called Cyclops. A DNA-binding transcriptional activator orchestrates symbiotic root nodule development. And the authors are Sylvia Singh, Katya Kotzer, Jane Lambert, Marion Cherry, and Martin Parnisky. They're from the University of Munich in Martinsried. So Cell Host and Microbe. Cyclops is all in capital letters. And this is a paper mostly about the plant host. And I know this is not This Week in Plants, but it's an interesting interplay between bacteria and plants. And I learned a great deal preparing for this paper, which is good. I think it's a really neat paper. This paper has to do with legumes, which are a kind of plant. And many of these legumes have symbiotic bacteria in their roots. There are these structures called root nodules that form, and the bacteria fix nitrogen from atmospheric nitrogen, and that's so the plant can make amino acids out of that nitrogen. And you'll know a lot of legumes like alfalfa, clover, peas, beans, lentils, soybeans. Even wisteria are legumes.
But the most famous is what most graduate students live on: peanut butter, peanuts.
It's the most famous legume?
Well, for graduate students, peanut butter is the staple of the starving graduate student. At least it was when I was a starving graduate student.
It's now ramen noodles.
Yeah, that's probably true.
So legumes fix nitrogen. The bacteria in the nodules are called Rhizobia. And our former TWiMer, Joe Handelsman, is interested in these bacteria. They also have fungi that are symbionts of many land plants. They're called arbuscular mycorrhiza fungi. And these guys acquire phosphorus for the plant. And of course, in turn, the plant provides them with a nice place to live. Do you know if the plant provides anything else to the bacteria? or the fungi? Does anyone know?
I don't know.
A place to live? They must. Seems like a place to live would not be sufficient, right?
I think if I'm remembering right, I think it may give them some sugar too.
Could be. So the cool thing here is that when the bacteria or the fungi encounter the root, this stimulates the development of the root nodule. So the root normally doesn't have a nodule, and then when it contacts the bacteria or fungi, well, the bacteria in particular make a nodule and develops into a structure, and the bacteria get inside the structure. And I found a very cool review article just a half hour ago in Nature Reviews in Microbiology. It's called Speak Friend and Enter, Signaling Systems That Promote Beneficial Symbiotic Associations in Plants. It's by Gilles Oldroyd. You guys know what Speak Friend and Enter is from?
I don't.
Not me.
Gosh. No, I can't
When
believe
you tell us,
Pop
we'll
culture,
probably
what is it?
immediately.
It's not pop culture. It's from The Lord of the Rings.
Oh.
Which is quite an old book. Well, not really old, but yeah, that's the thing that Gandalf says to get in to some the cave where the dwarves used to live. Speak friend and enter, and the door opens.
You know, if we were on the Big Bang Theory, we probably would have known that. But since Michele and I are neither on the Big Bang Theory, we didn't know about that.
Anyway, it's cool because the bacteria speak friend and the plant lets them enter. They have a great picture here. So the cool thing here, what happens is the bacteria, first of all, the bacteria are actually attracted to the root because the plant wants the bacteria. They make flavonoids that attract the bacteria. And then the bacteria come and they make what are called nodulation factors or nod factors. And these are chito-oligosaccharide backbones with sugars attached to them. Really interesting molecules. And the nod factors are detected by receptors on the surface of the root cells. Okay, and these induce the formation of the nodule. What happens is this root hair comes out. Well, there's a root hair already. The bacteria penetrate it, and they go into the root structure, and then this nodule is formed. That's really neat. Really,
Exactly in the right place, right where the bacteria are.
Yeah, and the fungi do similar things. They have chemicals that attract them, and they get inside the root also. They don't make a nodule, but they make an arbuscle, which reminds me of Arbuckle, but it's not the same thing. I guess you guys know what Arbuckle is, right? He was an actor, no?
Mm-hmm. And his first name was Fatty.
Fatty Arbuckle, right? I was forgetting.
Yeah. And it is indeed, the plants do indeed provide the sugar that actually facilitates the nitrogen fixation.
Okay. So how this happens is the subject of this paper. What happens is when the bacteria get into the cell, into the root structure, there are oscillations of calcium levels in the nuclei of these cells. So calciums go up and down. Calcium levels go up and down. And this is believed to activate the expression of genes that are needed to build this structure. They're called symbiosis-related genes. All right, so calcium going up and up and down, and that's linked to the induction of this structure. So how does this work? Well, of course, plants are eukaryotes, so they have proteins that bind calcium called calmodulins, and there is a central protein in the root called calcium and calmodulin-dependent kinase. CCAMK. There you go, Michael. Another abbreviation. Mm-hmm. It's a central regulator of root development. If you make mutations in this gene encoding this calcium and calmodulin-dependent kinase. So as the name would suggest, this protein is an enzyme that can phosphorylate things. That's what the kinase means. It's calcium-dependent, so it needs calcium probably bound to it to phosphorylate. And also needs calmodulin. And the way this usually works, calmodulin is kind of a messenger. So calcium is a signaler. And the way it signals in plants and also in vertebrates is by binding calmodulin, and then that interacts with some kind of sensor.
It's almost like the cyclic AMP catabolite activator protein in terms of activating the positive transcription factor in the lac operon.
Exactly. So there's a small molecule binding to a protein, right? Mm-hmm. And that's doing the actual thing. So this CCAMK is a really important protein here. In this protein, there's an autophosphorylation site, so it can phosphorylate itself and make itself active. And you can make changes there so it's always active without being phosphorylated. And then if you express this protein in plant roots, it will induce the formation of the nodule without calcium spikes. Okay, so the idea has arisen that the calcium spikes lead to autophosphorylation of this CCAMK, which then turns on other genes, right? Because, if you make a constitutively autophosphorylated calmodulin kinase, then the nodule forms without the calcium spikes. And again, the calcium spikes come from bacteria signaling to plant root cells.
So thinking about it from my simple background, it is indeed very similar to the way cyclic AMP and catabolite activator protein can, once it's activated, it can activate a suite of genes within E. coli. And similarly, this is what is presumably going on in the development of this very complex interkingdom. This is truly a bridging devices organ that's being created.
It's interkingdom. That's right. It's a common theme. You have activator proteins that turn on many other genes. And as you said, there are lots of other genes involved. So whatever the signals are that turn on the development of this root nodule, they depend on some transcriptional regulating protein. So in eukaryotic cells, we have DNAs can be transcribed into messenger RNAs at what's called a promoter. And the promoter is just the sequence that binds RNA polymerase. But there are many other proteins that interact at this site, and they can regulate transcription up or down. And in this case, there are two proteins called NSP1 and NSP2. These are proteins that are involved in nodule development. Then there's another one, a transcriptional regulator called NIN, N-I-N. It's not 9 Inch Nails. You guys know what 9 Inch Nails is?
You are full of the references
pop
today,
culture?
aren't you?
I'm a young guy.
He gets to ride the subway to work, so he has much time to read.
I just stare in the subway.
Oh.
But I know 9 Inch Nails. It's a rock group. Terrific. Trent Reznor. Yeah, he makes the whole thing himself on a computer. Anyway, I digress. So this NIN actually stands for nodule inception. NIN is a transcriptional activator that turns on other genes, okay? And then those genes are presumably involved in differentiating into a root nodule. Okay, so you have calcium activating CCAMK, and then some genes get turned on, and these include NIN and its targets. So the question is, what's the links between CCAMK and turning on these genes? So what they did in previous work was to say, let's find proteins that interact with CCAMK, and maybe one of those will be the one we're looking for. And that is what Cyclops is. It is a nuclear protein that interacts with CCAMK, and it's essential for symbiosis development, the development of the root nodule. If you make mutations in Cyclops gene, the plants do not make root nodules. If you give them tons of Rhizobia, they don't make a nodule. So this is essential.
So one of the things,
but the root hair curls, forming this unusual structure that looks like a one-eyed Cyclops.
Is that right?
Yeah, that's where the name came from.
Huh. Oh, I didn't know that.
Yeah.
I was looking for I thought it was an acronym.
No.
That's pretty cool. Thank you, Michele. So Cyclops is and of course, Michele, from that we get the name of this episode, which is eyeing root nodule development, right?
All right.
So Cyclops is a coiled coil protein. What this means, coiled coil proteins are often involved in regulating transcription. They have two alpha helices, which are in turn wrapped around each other, and they're often dimers. And then one end can interact with DNA. and regulate transcription. So this suggests that Cyclops is a transcriptional regulator. They also have known before that Cyclops is phosphorylated in vitro by CCAMK, and that's where this paper begins. So they start off simply by identifying the phosphorylation sites on Cyclops. They mix it with CCAMK and a phosphorus donor, and they do mass spectroscopy, which is now how everyone looks for modification of proteins. It's so powerful.
In the olden days, you ended up glowing in the dark with radioactive phosphorus.
Yeah, you would do that, and you chop it up into bits and try and see what part of the protein was labeled. Yeah, you would be really radioactive. That's right. But now you make a little bit of the protein, you hand it to your mass spec facility, which you guys all have, right?
And then you write a check.
You write a big check.
You write a very large check. Yeah, every school's got at least, that they're ubiquitously distributed, and in fact, even small 200-bed hospitals these days are MALDI-TOF has become distributed in them as. And so this is a common technique today, but you write a big check.
Yeah, it's expensive. So have you guys used mass spec in your work?
Not personally.
We've tinkered with it with looking at some of the MRSA isolates that we've gotten out of the clinical samples that we've been talking about. And we'll talk about that in the questions.
Yeah, that's right. So basically with mass spec, it will not only tell you the sequence of your protein, which for Cyclops was known, but it tells you how each amino acid residue is modified. In this case, they're looking for phosphorus, but you could look at methyls or acetyl groups or whatever and get all kinds of information about how proteins are modified. It's really, really very powerful. So they find 5 phosphorylation sites. Now, to get Cyclops phosphorylated, they add both calcium and calmodulin along with the CCAMK. All right, so... And as I said, calmodulin is the calcium-binding protein. So this is an important part of CCAMK's phosphorylation activity. You have to have calcium present in calmodulin. And that makes sense because CCAMK is sending a signal somewhere when it sees these calcium oscillations that are, again, produced by the bacteria coming into the root. So these 5 phosphorylation sites, the obvious question, do you need them for symbiotic development, for the development of the root nodule? So they take each of these 5 sites and they make amino acid changes so they could not be phosphorylated. They put the gene in plants and they see if root nodules form. So you can do this with plants. You can introduce genes. You can introduce all kinds of altered genes and so forth. And they have a nice assay. They just have bacteria there near the root. They grow the plant and they pull it out of the dirt and they look for root nodules. Maybe there's not even dirt. Maybe it's just growing in liquid. I don't know. But they put it under a microscope and you can count nodules. So 3 of these phosphorylation sites are dispensable for nodule formation and 2 are essential. And they also do this so this is done in plants. And these altered proteins, by the way, the altered Cyclops, are all in the right place. They're in the nucleus of the plant cell. They bind CCAMK, so it's not any of those things that are...
They're not screwing up the protein,
yeah,
so
exactly.
It's doing what the protein's supposed to do, but it's just not... The phosphorylation is the active player.
Right. So a cool part of these experiments, which is going to come up again, is the use of aspartate as what they call a phosphomimetic. Right? So,
cool term.
Yeah, the putting a phosphorus on an amino acid gives it a certain chemical signature, the phosphate group, and it turns out the side chain of aspartate is very similar in chemical properties to a phosphorus group, and so if they put aspartates at these 2 important positions, that will allow root nodule formation in the absence of Cyclops, of CCAMK. So Cyclops with 2 aspartates looks like, to the plant, it looks like it's phosphorylated, and you get root nodule formation.
So it does the effective thing of when that phosphorus atom binds to the Cyclops protein, it folds it into a different shape so that it has then allosteric consequence of the transcription biology. Yeah. And the aspartic acid substitution does the same thing with phosphorus. But how do they turn it off then?
Well,
They don't.
You can't.
It's always on.
Constitutive.
It's always on. These guys, in these plants, they make lots of root nodules. And if you don't have bacteria around, they'll make root nodules. They just don't have bacteria in them.
Which are presumably filling up with sugar, so then that would be selectively disadvantaged to the plant.
Right.
If you're not making nitrogen.
Right. Of course, in the lab, it doesn't matter. Right.
No.
So out in the field, that would be a problem. Yeah. So their idea now is that, as Michael has said, Cyclops transcriptionally regulates. And their idea is it's regulating this 9-inch nails promoter, the NIN promoter.
That's easier to remember than NIN.
NIN. NIN is, what is NIN? Nodule inception. It's not a bad word either, nodule inception. So Cyclops is phosphorylated by CCAMK, and that's in response to calcium, which is in response to bacteria. So there's a little pathway developing here. And then Cyclops phosphorylation regulates the NIN promoter, and then that turns on genes that are going to make the root nodule. All right, so this is what they want to look at. And now a lot of the experiments involve taking this NIN promoter, which is just a small piece of DNA, and they put a gene next to it that can be readily assayed. They use luciferase, I think, in this paper. They would call that a reporter. And then you say, can Cyclops activate this promoter reporter piece of DNA? And they find, in fact, that Cyclops will activate it when it's phosphorylated. So you can, if you add a Cyclops protein that's been phosphorylated by CCAMK, it will turn on the NIN-driven gene. If you use Cyclops with amino acid changes that prevent phosphorylation, it doesn't turn on the gene. And of course, if you use the aspartate substituted Cyclops, it will turn on the promoter. So it confirms their idea. Cyclops is turning on this promoter. So how is that working? They take the NIN promoter and they do what's called a Racan check. They just mutagenize it extensively, and they end up finding a 30 base pair piece of DNA that responds to Cyclops and will turn on a reporter gene. They call this the Cyclops response element. CYC is another abbreviation.
There's plenty.
Cyclops response element. All right. So typically these elements bind protein directly, and they confirm that. They show that they use a technique called electrophoretic mobility shift assay, which is quite an old technique we used to use years ago. I didn't know people still did this.
When I was a postdoc in the dark ages.
You basically take your 30 base pair piece of DNA, you run it on a gel, it has a certain mobility in the gel. And if it's binding to a protein, the mobility is less. It slows it down. That's why it's called an EMSA, electrophoretic mobility shift assay. And they show that Cyclops binds this 30 base pair, a piece of DNA. They also do some cool fluorescence experiments in plants showing that Cyclops binds the NIN promoter in plants. And then finally, in order for Cyclops to bind this 30 base pair piece of DNA, it has to be phosphorylated. And they have all these nice mutants of Cyclops that can't be or are constitutively phosphorylated, the phosphomimetic, and all of those show that phosphorylation is needed for binding. Okay.
And this was an important experiment because it shows that Cyclops acts directly by binding DNA.
That's right.
So they don't need to invoke, 2 or 3 other proteins in between Cyclops and the DNA promoter.
That's right. There could be other things involved there. And this is directly
so they can really focus their work now.
It's a classic case of a true transcription factor.
It is. It's a really classic. And in fact, it really helps that yesterday I gave a lecture on transcription in my course because this all came right back to me.
And you talked about electrophoretic shift assays.
Actually, I didn't do that, but we did talk about the structure of promoters, and we talked about it from a viral viewpoint because many viruses encode transcription proteins that regulate transcription. And so it's quite good.
It's one of the hardest things for students to wrap their heads around is this whole notion of positive and negative control, especially in a case like this Cyclops protein, where it has to be activated before it can actually facilitate its transcription activation.
Right.
So before it can bind.
Before it can bind.
That's right.
And when I talk to the dental students. I just did this a couple of weeks ago. I said, the way you keep things straight is if something has to be added to the target in order to see the effect, it's positive control. And if something has to be removed from the target, then it's negative control. And if you can keep that straight, then you can have all these variations on the theme of additions or subtractions to the effector molecule that is actually making contact with the nucleic acid. That's because you can have transcriptional control, and you can also, at least in bacteria, have translational control where the protein is actually binding the RNA. And in today's world, we have RNA that can interact with RNA, or RNA that can interact with DNA.
Right.
And so what you learn about transcription factor biology, you can actually take to those next levels where you're looking at all these new molecules like silencing RNAs and activating RNAs and all of these other new and cool things that are coming out of labs today.
So one of the things I talked about yesterday is the fact that many proteins that bind around the promoter and regulate transcription, they're often active as dimers, and they have multiple domains. They're typically DNA binding domains, and that's separate from the domain that allows dimerization, and that's separate from the activation domain, which interacts with other components of the initiation complex. So they go through the Cyclops protein and identify these different domains. They identify a DNA binding domain, they find the activating domain, and again, they just produce these individually and show that it'll either bind DNA or activate the promoter. They find that the protein forms dimers, and interestingly, the dimer formation is not affected by phosphorylation of Cyclops. Remember, Cyclops can be phosphorylated, and that's important for root nodule formation. What they do find is that binding of the Cyclops to DNA is much stronger when the protein is phosphorylated. So their idea is that the N-terminus, the amino terminus of Cyclops, which contains the phosphorylation sites, is probably masking the DNA binding domain until it's phosphorylated, and that makes a conformational change in the protein, so the N-terminus moves away from the DNA binding domain, and then the protein can bind DNA. So that's why phosphorylation is important, so this protein can bind DNA. So it's a little cascade here, really. And I don't know if I've mentioned this, but a lot of these experiments are done in a plant called Lotus japonicus. There's a pretty little plant here. You can go on Wikipedia and see all about it. It's a wild legume, and they use it in labs because it takes only 2 to 3 months to grow up from whatever they start with. So it's easy to study. It's got a pretty small genome, and you can manipulate it genetically. So they do a whole lot of studies in L. japonicus, for example. They show that if you synthesize Cyclops aspartate, this phosphomimetic of Cyclops DD we'll call it. If you put that in plants, it triggers the induction of a CYC reporter gene. So in other words, you take your CYC is the DNA binding sequence for Cyclops with a reporter protein gene attached to it. Put that in plants. If you then put Cyclops DD in those, it turns on the reporter. That turning on of the reporter also works in a plant where the CCAMK is not functional.
And sterile without the bacteria.
Right. No bacteria needed for this. Right.
Well, that's the classic proof that it's actually doing what it's supposed to do.
Right.
Downstream.
Yeah, they also have mutants of CCAMK, which are autoactive. I mentioned those originally. They're already active, and in other words, they will phosphorylate Cyclops all the time, and this, of course, turns on the reporter. If you have a Cyclops plant mutant, then this induction of the reporter doesn't happen. So all this information shows you that Cyclops is a key player in this pathway that begins with CCAMK, calcium calmodulin kinase. Next, they show that in roots where they're expressing or producing Cyclops DD, the NIN gene and its target, which is NFYA1. It's a gene turned on by NIN. NIN is the promoter, and NFYA1 is one of its targets. These genes are turned on when you put in Cyclops DD. So it's showing that it's upregulating the synthesis of downstream genes. And a couple more experiments. The CYC reporter, again, the DNA binding piece of the Cyclops site with the reporter is activated in the plants and roots that are inoculated with Rhizobium loti, which is one of the symbionts that can get into these roots, but not a reporter with mutations in the DNA binding sequence that we know prevent Cyclops binding. All right, so adding the bacteria ends up, you turn on this reporter, which is turned on by Cyclops, but not if the reporter DNA has been changed. And if you have a mutant plant, again, without Cyclops, you add bacteria, there's no induction of the reporter.
So, Michele, do you think that on Figure 6, where they show that beautiful picture of the GFU wild type, the effect of, I think what it is, a green eye, that's what Cyclops is all about?
No, because that's the wild type phenotype. So it's the Cyclops mutant phenotype that is quite striking. And Cell Press did an announcement in the front of the journal, and it showed a photo there. So there is a link to that on the TWiM page.
Yeah, I wasn't able to access those because we don't have a subscription.
Yeah, that was my problem. We're not as well off as Michigan.
Michigan is just really well off because of its sports teams, you know.
That's not true.
And it's science.
Our loyal alums. But I'd be happy to put a photo onto the website.
Yeah, I'd like to see that Cyclops. That would be cool. So I don't know if they say this, but they make plants with that lack CCAMK, but they make Cyclops DD, and they will make, Michele said this, they make root nodules without bacteria. Mm-hmm. So in other words, you don't need CCAMK if you got a phosphorylated mimic Cyclops. And then the last experiment is very cool. So now they know a number of genes that are involved in this pathway of root nodule formation. And they take separate plants with changes in each of these genes, mutations that knock out each of these genes, and then they put Cyclops DD into these and say, do you get root nodule formation without bacteria? Because you have the Cyclops DD, you don't need bacteria there. So you'll get no root nodule formation in mutants of this protein NSP1-2, NSP2-2, and NIN. So these are downstream of Cyclops. So Cyclops is activating them. Okay, so again, if you have a constitutively active Cyclops, which is binding the NIN promoter, NIN and downstream genes are essential because those are doing the work. Upstream genes don't matter. So there's a protein involved in calcium spiking. It's called SYMRK, SYMRK. And mutants of the protein, if you put in Cyclops DD, no problem making nodules. And there's another protein called Cerberus, Cerberus. This is a protein involved in getting the bacteria through that root hair into the root itself. That doesn't matter if you express Cyclops DD. And again, these are upstream of the Cyclops, so they don't matter. Normally they would be, Cerberus and SYMRK would be feeding into Cyclops, but since Cyclops is already activated, it doesn't matter. So it's pretty neat. So the model in the end, basically, CCAMK and Cyclops, they feel are bound together in an active complex in root cell nuclei. When bacteria initiate calcium spiking, CCAMK is activated by calcium bound to calmodulin. This phosphorylates Cyclops, which releases the DNA binding domain. It can bind the NIN promoter, turns on NIN expression, and then activates. And NIN is a transcriptional regulator itself, which turns on other genes involved in root nodule formation. That's pretty neat.
Well, I think what that shows is you have a transcription factor activating a transcription factor.
Right.
And so this really illustrates how complicated something like a root nodule can actually be. And I was really impressed in how they dissected it, the whole upstream and downstream, and actually driving home the importance of Cyclops and the phosphorylation events, because there are upstream consequences, or it's not really upstream. It needs to happen before Cyclops. And
that's right. That's why you have to have a sequence of events, because things have to happen at the right time, right?
And to expand on that, you don't want them to happen, you don't want it to misfire.
Yeah.
So often in biology, when the ultimate decision is a big commitment, a big developmental switch, there are naturally layers of signals, much like the telephone game that maybe you played as birthday parties as a kid. You want to make sure that the signal is robust. So by having multiple kinases in a row, it makes sure that the signal is strong and the cell won't make that commitment unless they really need to.
Yeah, yesterday in talking about viral transcription, there are a number of big DNA viruses that have transcriptional cascades. You know, the virus gets in, you make a protein, which is a transcriptional regulator. It turns on another set of proteins, one of which is a transcriptional regulator. That turns on DNA synthesis. And then finally, you turn on structural protein production later, because you don't need that early. So it's a matter of timing, making the right proteins at the right time. And probably that's part of the issue here as well.
Right.
In addition to making sure that you got the telephone message correct.
Mm-hmm. I wanted to add that we admired their gel shifts that showed that the Cyclops binds directly to DNA, and it looks so obvious. But I was able to communicate with two of the first authors, Sylvia Singh and Katya Katzer.
Nice.
And they told me that purifying Cyclops from E. coli was a real challenge, keeping it soluble. And then Katya, who was in the cold room for hours and hours and hours trying to get the gel shifts to work, said she had to do a lot of troubleshooting. But when she finally figured out the right salt concentration, it generated these beautiful figures that we're all seeing.
I bet they never heard of TWiM, right?
I didn't ask that, but they were both quite excited.
Cool.
And they told me a little bit about their backstory. So Sylvia is currently now, she's actually gone on to do a postdoc. She's continuing this line of investigation with Gilles Oldroy, who, Vincent, you mentioned his Nature Review piece today. So that's the same lab where she's gone now. But she got her start as a med tech and worked in a hospital looking at blood tests for a number of years. And then when she was moved into the pathogen side of the med lab, she got really excited about microbiology and decided to leave her job and go to grad school and start studying symbiosis. So that was her path. And then Katya, I'm sorry, Elio's not here. She wrote that she is passionate about picking mushrooms. She also has a biochemistry background, and that's what led her to study plant symbiosis. So she worked as a diploma thesis student under Sylvia's direction. The two of them worked as a team, and now Katya is continuing this project as a PhD for her PhD thesis.
Nice.
Do you think the mushroom picking thing is a European issue as opposed to a New World issue, because the Europeans have more empiric knowledge passed down from mother to daughter or father to son of what's safe to eat, whereas the New World, because we're often only one or two or maybe three generations into the U.S. or the New World, we don't know what's safe to eat?
Well, I also think, and this is a gross generalization, but in the U.S., we pride fast food, and in Europe, they really appreciate, food of the earth and how it's different in different regions, and they have a much more respect, I think, for food than we do.
Our idea of European food is the Italian restaurant, the German restaurant.
Yeah, well, we have some people who appreciate good cooking in this country.
Absolutely.
In fact, there are foodies everywhere.
Yeah, it's gotten much better from. I helped my son do an essay. He had to write about the changes in eating in the U.S. since Fast Food Nation was published 10 years ago. There have been, cooking is now a game show thing. People, go on contests and cook well, and There have been a lot of changes. It's still a big problem. The fast food industry has ruined agriculture in the U.S. in many ways, but it's getting better. Anyway, it's a nice paper. I really enjoyed that. So Sylvia and Katya
the data are beautiful.
And Jane and Marion and Martin, thank you very much. We hope you enjoyed our discussion. And if you find anything we missed, let us know. And I would like to read some emails. Here we have a few backed up. So let's start with Mark, who writes, "Hello, Team TWiM. I followed with interest your coverage of Michael's research into the use of copper to fight hospital infections. Of all the interesting papers covered in 2013, I think the one most actionable is episode 55, The Copper Room. His research, described in that and prior shows, addresses an underreported aspect of our healthcare system. Hospital-acquired infections are a serious matter, of which I am especially mindful as I prepare for surgery later this month. Kudos to Professor Michael Schmidt for proving how a relatively simple change of hospital materials can have a huge impact on improving patient health and outcomes. Let me share some news and ask follow-up questions. Monday, 1/6/14 at the Consumer Electronics Show, Corning Incorporated introduced a new line of Gorilla Glass that it claims is the world's first antimicrobial cover glass, and he sends some links to this. Corning is positioning the product for touch-based mobile devices to combat bacteriological growth and spread of disease. We have all seen phones whose surfaces are in plain speech gross, disgusting, and covered with layers of schmutz. TWiM listeners would call such objects fomites, to use a polite, abstracted, and neutral term.
This is true.
Here come my questions. 1. Corning highlights the use of ionic silver as the antimicrobial agent in its new glass. Silver Ag is in the same column and one row below copper in the periodic table. Below silver is gold. Do I recall my chemistry correctly in that each has the same number of valence electrons? If this is true, can Michael compare and contrast how copper, silver, gold vary in effectivity killing bacteria? Besides economics, is any one of those metals a preferred choice for bacterial killing agent? Why and why not? Is the preference related to their increasing conductiveness as you descend rows in the periodic table? All right, we should stop there and let Michael.
He's got
that.
many interesting questions. First, thank you for the compliments, but research is a team sport, so my colleagues who assisted with that work, Cassie Salgado, Joe John, Bob Canty, Ken Sepkowitz, all should be commended for putting that piece of work together. Clinical trials are never easy. But as to your question, I too was absolutely fascinated by the Corning announcement, and it's interesting to start with the simple question, why copper, why silver, why gold? Economics is a big issue. Copper is, of course, priced at the pound, while silver and gold are priced at the troy ounce. So that's number one. Number two is copper, from a fomite perspective, is better at room temperature. Its antimicrobial activity is effective at typical temperatures that we keep for conditioned space, typically 15 to 25 degrees C. And while silver, in order to have the same antimicrobial potential as copper, needs two special things to happen. You need high humidity, typically 95% relative humidity, and temperatures exceeding 37 degrees in order to have the same time to kill that copper does. That does not necessarily say that the silver in the Gorilla Glass will not work. It just simply says it will not work as quickly as the copper. And the other issue is I went and read the disclaimers that were associated with the Corning piece, and they are only getting the treated article exemption from the US EPA. They don't have the ability to make public health claims as yet. So that means the bacteria won't eat the glass, is what effectively a public health claim, a treated article exemption. The silver in that product will prevent it from making an odor from the bacteria associated with the glass. It doesn't necessarily mean it will be sufficient to kill the bacteria. From what I understand from Corning is they're currently putting that forward to the EPA to actually validate, and they're hoping to get public health claim, I think is what I had read. So as to the mechanism, the data that are in the literature are associated with dry copper. We have to divorce 50 years of prior literature that was principally looking at ionic copper, which was in the form of typically copper sulfate or copper oxides. And we really need to look at the metallic variants. And the data that we did a TWiM paper by Warnes last January about how solid copper can prevent the conjugal transfer of multi-drug resistant from Klebsiella to E. coli and vice versa. And that was by Warnes and Keevil. And they have done an elegant series of papers really going to how the mechanism of action of copper is inactivating a bacterium. And principally what happens is you're right to think it's about conductivity, but remember, it's all about the electronic potential of the membrane, the energized membrane of the bacterium, where the copper facilitates a cascade of irreversible events leading to the rapid death of the microbe. So what happens is when the microbe is sufficiently close to the extremely partial positive charge of metallic surfaces like copper, the bacterium will actually donate its electrons to the metallic surface as a consequence of the high flux required by living cells. And flux simply means electrons moving through the membrane. And when the bacterium loses its electrons by this literally shift of electrons to the copper metal, because the electron and the proton are an uncoupled event in the bacterium. Remember, the proton is ejected independently of the electron, but they must come back together at the end of the membrane chain in order to facilitate charge balance inside the cell. And so the consequence of the copper taking an electron is you collapse the membrane potential. And when you collapse or dissipate the proton motive force, and this has been observed over and over through the use of these redox-sensitive dyes by Keevil and Warnes, where they actually show how quickly the membrane potential collapses in the presence of metallic copper. So I'm not just hand-waving saying proton motive force collapses. Keevil and Warnes have actually developed this technique and actually can measure how quickly the membrane potential collapses. And then in the MBio paper that we discussed last January, Warnes and Keevil showed that not only does it collapse the membrane potential, but then you get the concomitant destruction of the DNA as a result of the free radicals that are generated internal. When you collapse the membrane potential, because the proton can't come back in because there's no electron, you get the peroxidation of the membrane, and you generate more peroxide inside the cell, which then cleaves the nucleic acid. The mechanism of silver, because silver passivates, or anyone who's ever had to polish silver for their mother or grandmother before Thanksgiving appreciate that silver tarnishes. And this passivation prevents this ability because it sets up a barrier. And it's all about proximity of the microbe to the metallic surface and that strong partial positive that the metal is exerting that results in the collapse of the potential. And I think that's how it's working. Now, in the case of this Corning glass, where you have electronics underneath, they may actually be seeing something where the electronics are facilitating this potential generation. And so Corning may be seeing something that is, they'll actually be able to kill bacteria. So I'm waiting with bated breath to see what actually happens.
Is the removal of electrons from the bacteria have to do with the valence electrons of the silver and the copper?
Well, it's metallic copper. And if you talk to your quantum mechanic friends, it's a metal has all of its valence electrons, even though it's electrically neutral, available at all points in time. So it's effectively how you get flow of current.
Got it. And is gold antimicrobial as well?
Gold is noble. It's a noble metal, and so the bacteria just ignore it.
Okay. So you went through the mechanism. What impact does copper have on neutralizing viruses?
It, again, Keevil has published just recently that it can inactivate norovirus. He used a mouse variant of norovirus to demonstrate it can inactivate it. He's previously published that it can activate influenza. And my gut suspicion there, it's a completely different mechanism where it may be destabilizing the structure of the virion and exposing the nucleic acid to the rigors of the environment.
That's interesting because for noroviruses, surfaces are implicated in contamination of food, for example. So that could be very useful. That could be
huge.
Yeah. And Mark says, I use neutralizing because you can't kill a non-living virus. Do you agree with this phrasing? Yes, I absolutely agree. Wishing you all the best in 2014, Mark. And P.S. The weather here in the San Francisco Bay Area, the weather these past few days was mostly sunny, highs in the mid-60s, lows in the mid-40s. This East Coast refugee empathizes with Easterners like TWiM's Michiganders, Carolinians, Jerseyites, or his brother out on Long Island, who have been experiencing extreme cold with daily highs in the low teens. Only Alios San Diego has better weather as I write this. Hey, Michele, he called you an Easterner.
We're in the Eastern Standard Time Zone, so...
I just want to know if that's okay with you to be
Sure.
called an Easterner. Where are you from originally again? I forgot.
Well, my parents are from Sault Ste. Marie, so way up in the northern reaches of Michigan.
So
you're
you're
back
originally a Michigander.
I have roots here in Michigan, that's right.
Did that influence your going to Michigan?
Well, you know how it is when you're a postdoc, you apply to the jobs that are available. And I was delighted that there was one here at Michigan.
So you went there out of a postdoc and you've been there ever since?
That's right.
Okay. All right, the next email is from CN, who writes, on the foremost, thank you for an excellent educast. Your podcast has helped me generate and understand a lot of ideas, which I would not have never accomplished had it not been for your podcast. I've just joined the Department of Neuromicrobiology in Bangalore, India, as a PhD scholar. Maybe I can consider it a Twix bump. Sure. The issue of healthcare-associated infection has come up multiple times in your podcast. I have a query in connection with the same. Hospital environment harbors microbes, usually of the MDR type, multiple drug resistance. The ones commonly involved in nosocomial infections are often environmental and antibiotic-resistant types. There is a very limited scope of competition from harmless and less robust environmental flora. I'm curious if we could theoretically at least introduce environmental flora into a hospital regularly, which would then reduce hospital-acquired infections. Just a wandering thought.
It's sort of the hygiene hypothesis enforced on fomites. It's not a bad idea. And, folks have played with it, but we don't quite understand whether or not that's going to place the immunocompromised or at-risk patient population at risk because, again, most of the time, something that, the physicians and the nursing staff and the technicians are walking around the hospital and they're not becoming ill. It's a bit more complicated, but people have talked about the hygiene hypothesis for the built environment.
Interesting.
But I would hope they also talk about the threat of plasmids and phage jumping from a environmental strain or from a pathogenic strain into environmental strain, and you have new pathogens emerging.
Oh, no, that goes without saying.
Yeah.
I mean, we have enough infectious disease physicians who have been burned by the promiscuity of our friends and, moving pathogenicity islands. The
Right.
operational definition of a pathogenicity island is the microbe that's the pathogen has the island, and the one that's not the pathogen lacks the island. And, that's effectively moved by phage or by conjugation or, in some rare instances, transformation.
So probably it will be a while before we use environmental flora in our hospitals as a preventative.
Yeah, we'll be using copper before.
Yeah.
We'll be using copper. Florence Nightingale would be spinning in her grave if we started to do that.
I've always wondered why TWiM doesn't have a pick of the week. Keep up your excellent work. Never stop, Twix. Another Twix podcast fan. I don't know, CN. I just thought to do differently with a different podcast, not the same thing with all of them. That's all. Next one's from Mark, who writes, firstly, thanks for the show. It regularly blows my mind and keeps me very engaged during the otherwise cold and rainy walks into work. Keeping up with each episode has very much broadened my mind to microbiological topics that I wouldn't have come across otherwise, which in turn enriched my own thinking and work. In the fascinating TWiM 68, The Fungus Among Us, Elio questioned the existence of uninhabited aqueous environments. I know of 2 naturally occurring habitats on the Earth that are probably functionally sterile, that is, supporting no active growth. These are Discovery Basin in the deep Mediterranean and Don Juan Pond in the McMurdo Dry Valleys, Antarctica. Both of these are extremely concentrated brines, where the main salt is not sodium chloride, but magnesium chloride in the case of Discovery Basin, and calcium chloride in Don Juan. Previous studies report DNA sequences from Discovery Basin, but the current thought is that these originate from organisms that live above the seawater brine interface and have simply sunk into it. mRNA has not been successfully recovered, suggesting that there is no active life. Exactly what property of the brine precludes life is not fully understood. It could be either the dramatically low water activity, availability of free water, or the chaotropic disruptive effect magnesium chloride has on biological macromolecules. Check out this paper, which discusses support for a chaotropic barrier to life. And he sends a link to a paper called Limits of Life in Magnesium Chloride-Containing Environments. Chaotropicity defines the window. Don Juan is a particularly fascinating place as it never freezes over despite experiencing temperatures down to minus 40 degrees Celsius. There is much less published biological data on this environment, none with molecular tools, but it is more chaotropic and more concentrated than Discovery brine, and most microbiologists who work with dry valleys lakes consider it a sterile environment. These environments are particularly interesting because of the current paradigm in astrobiology of liquid water being the holy grail for habitability. And yet, even on the Earth, there are, albeit rare, examples of aqueous environments that may fundamentally preclude life. It's neat. On a different note, I thought you might all enjoy this paper exploring some of the novelties of the haloarchaeal genome. This is a very cool paper.
I saw that.
Which says that if you take out all the origins of replication of DNA in the archaeal genome, the bacteria grow just fine. The archaea grow just fine, and
even faster than the wild type.
Yeah. So this they suggest that replication occurs by a recombination dependent process. So it requires a recombinase called RAD-A.
He also points out that particular species has multiple copies of the chromosome, so they can be a bit more flexible, perhaps.
Yeah.
Whereas E. coli with one copy has to have more regulation.
Yeah, this is a neat paper. We might do this. I'm going to take a closer look at it. I was reminded of it by your discussion of horizontal gene transfer in the archaea. It's a really nice study and highlights how different and interesting these organisms are when compared to bacteria or eukaryotes. The authors hint towards some profound evolutionary implications in the discussion. Apologies for the ramblings, and thanks again for all the inspiring discussions. No problem with ramblings. We do that all the time. Mark is at the UK Centre for Astrobiology at the University of Edinburgh. Neat. Astrobiology. I'm always amazed to find who listens to us. right? I think that's cool. The next one is from Bradley, who says, if you can find the backstory to this, it would be an interesting TWiM. And he sends a link to an article on CNN, passenger with possible tuberculosis infection pulled from plane. Now, this is a story that broke not too long ago and was covered extensively, December of 2013, a flight getting into, they're waiting at Phoenix Airport, and the pilot said, there's someone on board with TB and you all have to just sit tight while we take them off. They took them off, and they said everyone was probably infected. But I looked into this, and I found an article which said that the public health there in Phoenix tested it all and said there's no TB. That passenger did not have TB. There was no risk to anyone else. End of story. What I found interesting is that the health authorities tweeted out that piece of information.
Huh.
To everyone. No TB.
I didn't realize that the CDC communicates that information to the Department of Homeland Security, which in turn communicates it to the airlines.
I didn't know that, yeah.
Yeah,
because tuberculosis is a reportable disease.
Right.
And I wonder, I don't know when this came about, and I was going to call up my buddy at the CDC and ask him, is this a consequence of Spectre?
It is. According to this
plane.
article. Yeah, 2007, it's a new do not board list policy.
Yeah. All right. Next one's from Nathan, who writes, hello, Twimmers. A friend of mine just posted this question on Tumblr, and I thought you would be the perfect people to address it. And this is a question posted on a blog called Recursive Muffin, and it is, question of the day. A strain of Flavobacterium evolved the capacity to digest nylon, obviously in recent history. Fine and well. How long will it be until one of the karyogenic bacteria species evolves the ability to digest dental resin? After all, we are putting a lot of it on their dinner table. Michael.
So, we had our student dental research day today, and I was a judge of the posters, and I have been taking the copper story to the next level by looking at this, and I'll give you some backstory. First, this has already happened to some extent. In the United States, we currently spend $5 billion a year replacing resin-based composite fillings due to failure. The average lifespan of a resin-based filling that a dentist will put in today is around 6 years. And the recurrent decay usually compromises the restoration earlier in its lifespan. And that's when the bacteria are effectively going after the glue. And they're going because it's a polymerization, and the microbes, if the restoration isn't properly fixed and properly cured, there's enough carbon in there that they can get at before the polymerization is completely done, that they can actually get after it. And then afterwards, matrix metalloproteinases and cathepsins places the longevity ceiling at that 6 years, even in healthy and bacterial-free restorations. And so these matrix metalloproteinases and cathepsins, which are expressed specifically in dentin, they come in and they cause the restoration to fail. And
but
so
those are bacterial, Michael, or those are
those are eukaryotic. The bacterial failure. That's the places the longevity cap at around 6 years. So there's currently a resin-based product in the market that's from a Japanese company that actually puts chlorhexidine into some of these resin-based products in order to prevent microbial attack and to take out the it's principally to take out the bacterial attack. And so we've been looking at copper nanoparticles to effectively prevent some of this decay. But what he is hypothesizing has already been happening in the U.S., and it happens throughout the developed world, any place people are using resin-based fillings. The old silver fillings typically last 25 to 30 years without incident, and most people were concerned about the mercury issue, but the amount of mercury in an amalgam-based filling is insignificant in terms of health consequences. If you look at the evidence-based literature, it really has no issue associated with the health of the individual.
And I don't suppose there's any data saying whether the half-life of the resin is decreasing, which would be consistent with his idea that we've selected for bacteria. We're enriching for bacteria that can break it down more readily?
No, because the resins have gotten better. The polymerization agents and the curing times and so we don't have a clean experiment to do it. The folks haven't actually looked to see if there are resin-based dissolving bacteria, but that's a question that I can ask my friends at the Forsyth Institute, whether or not they've hunted to see if there are any resin-based creatures, resin-eating bacteria out there. But it's all about the polymerization, because the polymer needs to be perfectly cured. And any of you who've had a recent composite filling, you remember the dentist putting on the dark glasses and giving you a pair of dark glasses, and then they put the magic light into your mouth to cure the filling. And typically they only put the UV lamp in there for 20 to 30 seconds, and that's what starts the curing process.
Yep.
We've all made polyacrylamide gels, and it's a variation of polyacrylamide gels, except we use Temid and what's the inorganic, the inorganic salt. I haven't made a gel in.
I can't remember either. Oh.
Because you just pull APS,
ammonium persulfate.
Ammonium persulfate, and that's always goes bad. The binary catalyst.
All right, next one's from Darcy, who writes, "Hi, great podcast. I just wanted to let you all know that stool taking the shape of the container isn't a Britishism. I work in the micro department of a hospital reference lab, and we use this criteria on a daily basis when testing for C. diff. Specimens that are too formed must be rejected for testing, and we've had many discussions about what makes a stool formed or not. The criteria takes the shape of the container has turned out to be the best way to decide if we will reject a specimen or not."
Thank you for your service, Darcy.
That's
absolutely.
Yeah.
That was the name of that podcast. I really thought that was cool.
Yeah.
So I suppose Darcy is not in the UK. She did not tell us where she is, but judging from the email, not. And finally, let's do the last one from Jesper, who writes, "Dear Magi's of the Microbes."
Ooh.
I like that.
He must have been writing this around January 6th during the Epiphany.
In the news here in Sweden, there are unfortunately many reports about the spread of C. diff, and we've had a few deaths as well. Remembering Michael's research about copper surfacing as a means to reduce harmful microbes in hospital settings, it may be of interest to learn that a study has been conducted here to show that using chlorine and a detailed cleaning protocol reduced infections by 50%. I have not found the publication, but here is in Swedish the protocol recommended, and he provides a link. Essentially, they swipe all surfaces with chlorine. Here's another article about the research in a Swedish medical journal, also in Swedish. "I did not want to make the impression it was unknown that chlorine is effective as an antibacterial. CDC knew this," and he sends an article, a link to a CDC publication on this. "Thank you all for a very interesting podcast." What do you know about this, Michael?
Well, Clorox is the principal manufacturer of the bleach that's used in the hospital grade disinfectants. And typically in your household, effective bleach is diluted 1 in 10, and it generates an effective chlorine concentration that's the effective dose to inactivate bacteria. And we've, especially C. diff, and in the U.S., we all know about C. diff, and we all know about how effective good old bleach is, but folks don't like the smell. And again, as little as 3 parts or 3.5 parts per million can be detected by the human nose as odor, and we don't like the swimming pool smell in our hospitals. And 1,000 parts per million of that gas is likely to be fatal after a few deep breaths. And in fact, in the U.S., OSHA has actually limited your exposure to that odor to 0.5 parts per million in an 8-hour time-weighted average, where they consider a full-time job as being 38 hours a week. And so the CDC does indeed recommend bleach for disinfecting for C. diff. But Clorox is working on something as effective as chlorine-based bleaches that won't have the smell nor the wait time or the neutralization that's often required when you're using bleach. Typically, in the hospitals, they use it at about 1 to 5 in order to generate the appropriate parts per million.
Of course, for C. diff, person-to-person spread is also important, right? And
you can't do much remember the stool cloud. Remember the stool cloud that you're effectively walking through. And when you flush, this is another reason you should close the door when you're using the restroom, even at home, because of the cloud that will move, especially if your restroom is close to your kitchen. You want to control. And oftentimes when you're living by yourself or no one else is home, you often don't close the door to the restroom.
This is getting awfully personal.
Well, you know.
TMI? Is it TMI, Michele?
When you talk about these things to infectious disease professionals, and it's amazing some of the things that you begin to discuss and what you learn about people. You can just do a quick survey of your friends. If you're living by ask your graduate students tomorrow, Michele. Often, many of them live by themselves. Ask them, how many of you close the door to the restroom, and how close is it to your kitchen?
So the toilets make quite an aerosol when you flush them.
Oh, yeah.
Right?
Yeah.
Even home toilets, not just urinals in men's room make a huge aerosol, I'm sure, but toilets do.
So this is a new argument for putting the toilet seat and lid down.
When you flush, yeah, sure.
Women of the world will love me.
You know, I've been thinking about this lately because I learned at a podcast I did last year at the GM in Denver, talking to Bob Garcia, who works on polyomaviruses. We all excrete about 100,000 polyomaviruses per milliliter of urine. All of us, because they're replicating in our kidneys, and we're fine. But, you know, it's when you flush a toilet, you're aerosolizing that and inhaling it.
Mm-hmm.
So,
and that's how you get it.
Well, you get it very young, and so that may be part of it. But you just have to think about this, or maybe you shouldn't think about it, otherwise you'll go crazy. Maybe that's the solution. But now, whenever I go into a bathroom, especially a public one, it's one thing in your home where, you know, the flora is you. It's pretty stable, your family. But
right.
You go into a public restroom, and you can imagine the cloud as many people are flushing toilets. And it's pretty interesting that we're all as healthy as we are. We have good.
If it doesn't kill us, it makes us stronger, right?
That's right.
Build up an immune system.
That's another...
very knowledgeable.
That's another title. Anyway, on that note, we will end TWiM 73, and you'll be able to find this at iTunes and at microbeworld.org/TWiM, along with all the other episodes from all the way back to number 1, and they're all archived there for your listening pleasure. If you like what we do, one way you can help us is to go on over to iTunes and go to the podcast section and find us. Leave a short review or some stars, and that helps to keep us visible there. We love to get your questions and comments. Please send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thanks for joining us, Michele.
My pleasure. We've got a big basketball game this weekend.
Who are you playing?
Michigan State. So we're tied at top of the Big Ten.
Wow.
And we're playing them on our home court. So big implications.
Are you going?
And
I'm going. Yes, I'm going.
And it's getting close to March Madness.
It is.
Well, good luck with that game, Michele.
Thank you. And by the time this posts, the listeners are going to know the outcome.
Of course. But they'll be happy to hear that you went. You know, someone who was actually there.
Oh, yeah.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent. Thank you, Michele.
And, of course, where Michael is, there's no big basketball game coming up, right?
No, we're undefeated.
That's right. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiM, and Chris Kandayan and Ray Ortega for their technical help. The music that you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.